Welcome to the 2021 Hidradenitis Suppurativa Virtual Awareness Summit. Thank you so much for tuning in this year. Last year was such a blast, and I'm so glad that we were able to bring this to you again. This week is a very informative week filled with so much education, not only for patients, caregivers, and physicians, but also for just your average person who wants to know about HS. Because if you did not know, HS affects 0.01 to 4% of the population in the world. So that means millions of people across the world have HS or could have HS. So having your friends, your family, your neighbors, everyone educated about HS helps to lessen the gap of patients getting diagnosed within an average of 7 years. That's a long time to get a diagnosis and deal with something so debilitating like HS. So if you have someone that you think might benefit from being educated, make sure that you send them the link to sign up and register, because this is a place for everyone, and we are about educating everyone from top to bottom, patients to the general population, okay? So before we get started with the giveaway, I wanted to thank our sponsors, because this summit, just like last year's summit, was sponsored by not only foundations and community organizations that are supporting and driving advocacy and awareness for the HS community, but also small businesses. We love to partner with small businesses. And some of those foundations, community organizations, and small businesses that are sponsoring us this year are the Hidradenitis Suppurativa Foundation, the Mama IU, Hydraware, 100-page journals, designs by Muriel, HS Connect, Hope for HS, and Sugar Lovin'. So thank you so much. We are going to get started. We have Dr. Lynn here, who is going to be talking about HS and genetics. So I am going to allow her to give her introduction, and we are going to get into the presentation. Hi, Dr. Lynn.
Hi, Jasmine. Thank you so much for this opportunity. I'm really excited about this.
Absolutely.
I am hoping actually to get an idea from your attendees about what they're interested in about genetics. And so I don't know if there's a way to unmute, or if people could just put in the chat if they're here for a specific reason, if they have specific questions about genetics. That way I can make sure that I hit on everything in my lecture. Yeah, and so a little bit about my background. I have been in love with genetics since I first learned about Gregor Mendel and his pea experiments. I don't know if anybody remembers that from grammar school science classes. But he was able to, just by studying pea plants, figure out that genes must exist. And that idea of being able to just study patterns in living things and connect that to your DNA or to your genome has always fascinated me. I have worked in a DNA sequencing lab at Rockefeller University. I decided I wanted to run my own research studies and left to Columbia, where I completed my doctoral training, learning statistical methods for mapping genes or figuring out where disease genes are in the human genome. And my main interest for doing that is because if we can figure out the genes that are causing disease, it gives us some insight about the biology of the disease and can very often give us clues about how we can manage disease better. So before I get into my talk today, I don't have any commercial relationships to disclose. My research is funded just solely from the government and from Columbia University. I don't mind, I would actually prefer to be interrupted with questions as I'm going through this. Okay, so here it is. Here's the human genome. In every cell in our body, we have 23 pairs of chromosomes. So each, it comes in pairs because we get one copy from our father and one copy from our mother. So those are our two copies. Each one of these chromosomes has a different sequence, different content on it, has different instructions for different functions of the cell, but they're broken down into these 23 units. And so when we speak about the sequence of the DNA, it comes from just the molecular structure of it. Not so important. The main point is that there's 4 different, what we call base pairs that can join this double-stranded helix. So C, G, T, and A. And when we speak about the sequence of the human genome, it's just following along one chromosome. And what is the order of those bases? So this sequence is C, C, T, T, and it twists over, A, A, C, T, so forth. And so how does the genome work or influence us? It contains instructions for the cell on what proteins to make and how to respond to things in the environment. And so it contains the instruction to make these proteins, and then the proteins are actually the molecules that make up the cell, that send out messages from cells to other cells to communicate and coordinate. But all those instructions, they're encoded in this human genome. And so as I mentioned, so this is just the basic anatomy of a chromosome. When they were first discovered, we didn't have technology to actually read the sequence. And so geneticists talk about a gene being located on a short arm or a long arm. You can stain chromosomes with chemicals and you get this banding pattern. This banding pattern is the same in every human being. And so it also gave early geneticists a way to speak about a location in the genome. So they called each of these bands has a particular number associated with it. So for example, this is chromosome 1. So this chromosome here, this particular band, they call P32.3. And that's what's called the cytogenic location. As I said, more recently, we have technology where we can actually scan across every chromosome and read its actual sequence and know its physical position. And so you can start with the very first base pair at the end of the chromosome and just work your way down it. This chromosome 1, so these are in millions of base pairs that you're reading here, where a base pair is just one sequence, one letter. On chromosome 1, you have 249 million base pairs across this whole chromosome. And we do know the sequence of that chromosome. So you can go in databases online and just download the sequence if you're interested in it or read it. Okay, so what I mentioned is that here you have homologous chromosomes. So geneticists have a lot of like unique vocabulary that drives me insane because other people don't know what we're talking about. I try really hard to use regular words. So forgive me if I don't. So this homologous chromosomes, again, are just the pair of chromosomes, the one pair that you received from your father and the other one that you received from your mother. And so as you read across the sequence, most of the base pairs in all of our genomes are exactly the same. So if you go to this position on chromosome 1, all of us will have a T there, all of us will have a G next to it, a T, but then every now and then you come to a position that we call a genetic variant. And that means that at this particular position, you can have two different flavors, right? So some people will have a C at this position, other people will have a T at that position. And this is what we call a genetic variant. Geneticists call these two different alleles, but like I said, they're just two different versions. And so how frequent are these genetic variants? They can be shared by groups of people. So there are some variants in our genome that most people will share the same copy. So even though Jasmine and I have no relationship, we could both have a C at this particular locus. At the other end of that spectrum, every one of us has genetic variants in our genome that are unique to our families or unique to ourselves. So in that case, like Jasmine and I would not have the same sequence here, that this C might only be found in my family or only be found in me. So that'll be more relevant later when I speak about it. And so just to kind of review all of this terminology, we have a genome, which is the whole complement of DNA that's in every one of our cells. The chromosome is the way the genome is broken up into these smaller units. Let's see, you can have different kinds of genetic variants. Like I said, just a switch in the actual base pair. Sometimes in the genome, you'll get insertions. So you'll get a bunch of letters inserted where there used to not be any, or deletions where your genome will just drop a position in there. And so those are the different types of mutations. Not all of them affect proteins. So your genome is 3 billion base pairs. About 1% of that actually codes for a protein. And so here I'm just showing features, like as you go along a chromosome, you'll see in boxes here, these are sequences that actually will code for the protein, but you'll also have sequences that don't code for any protein. Sometimes they can be important because they can tell a cell how to turn on or turn off a gene, how to make a protein or not make a protein. And then sometimes the variants will actually change the protein itself and change the way it works. So I think that's just a really broad overview of what a genetic variant is or what a mutation is. Lots of them are in your genome. Not all of them affect proteins. Not all of them affect the way the cell functions. There's a lot of what we call benign variation, which means that there's a difference, but it doesn't cause any effect, so it's not important. Yeah. And so now I want to get to the point where Jasmine was asking about the difference between genetics and epigenetics, and somebody else had mentioned that too. And so if we think about every cell in our body has the exact same human genome, right? The exact same sequence in every cell of your body. But your body has over 200 different types of cells in it. So how is it that the same set of instructions can actually create all these different cells? And even among cells that are the same, it'll assign different jobs to different cells in your liver, for instance, right? And so how do all of those changes come into play from just one set of instructions? And that's what epigenetics is. Epigenetics accounts for those differences in the way a genome is expressed in action in a living thing. And so one way that you can think about it is you can think about the human genome as sheet music, and you can think about epigenetics as the music that gets played by the different people reading the sheet music. And so two different people can read the same sheet music and even be using the same instrument, but still make different sounds or make a melody that sounds different. And so you can think of that as epigenetics. The reason why people are interested in epigenetics is because it's easy. Your genes, basically, your genome is set from the time you're born. You have the same set of instructions. But as you go through life, you have you encounter different things in your environment, for instance, and those can change the epigenetics of your genome. That can change the way the music gets made or sounds. And so there's lots of opportunity then if you think about things in your environment that are making you sick, like the epigenetics is what is mediating those effects. And so sometimes people who are interested in studying diseases like me will study the human genome, and other people will study the epigenetics of it.
I think that's a good transition to this next question. So Jerome asked, does HS work like sickle cell or other diseases? Sort of like, for an example, can one parent have a trait but not the disease and pass it on to a child? And that kind of goes into the question that another person asked, which was, can you pass HS on, and I guess what's the probability if we know that?
So I actually have a couple of slides about that. Actually, that was a great transition to my next set of slides. And so, so how do you go from this like unique sequence, right, or this change in your DNA to a disease, right? And so for that, I do have to give a little bit of like biology background that has to do with the way cells divide. And so we know in our body, like our skin regenerates and you get cell divisions replicating and forming two cells that have exactly the same content as the parent cell. That is different when you're making gametes or sex cells, right? And so this is how eggs and sperm are made. And so in these cases, you don't get an exact copy of the genome. You end up with a cell that has half the genome, right? Because then during fertilization, you get one parent contributing half of their genome and the other half coming from your other parent. And so the cool thing that happens with this, so I mentioned that in your genome, you have one set of one homologous pair from your father and the other one came from your mother. But what you actually pass on to your children is a mix up of that. And so what happens when your body is preparing to make gametes, the homologous chromosomes, the pairs come together and they actually swap material. So it's like taking a deck of cards and shuffling it. So you start out with a chromosome that's identical to your dad's, but what you pass on is actually a mixture of this one chromosome having part of your dad's DNA and part of your mom's DNA. And so in that way, like in your kids, they're not all coming from like your paternal line or your maternal line. All of them are a mixture of all of their ancestors that came before them. And so this is just saying that same process again. And I'm just going through this. So when I talk about inheritance and disease genes getting passed through generations, it's important to know that like there's a lot of mix up that's going on between generations. And as a geneticist, we can leverage this to identify disease genes. And so as I mentioned, so this is, you know, in your genome from your parents, in your gametes, you have this shuffling of the cards. So you end up with these hybrids. And then in your children, they actually end up with bits and pieces from all four of their grandparents in their genomes. Now, how these cards are shuffled, it's exactly like shuffling a deck. It's random, right? And so because it's random, by chance, you could end up with the same piece of chromosome that a sibling has, or that one parent has, or it can get shuffled so you actually end up with, so you don't get like the same genome. And so this answers one of your questions. Yes, there are some diseases that people can get that are caused by a single disease mutation that's in the genome and can get passed along to offspring. And so here we can see here that in the grandfather, they have this disease gene. And during the process of meiosis, it gets shuffled up, but somehow this offspring still ended up with that disease gene. In the next generation, it got shuffled even more. So now this portion of DNA is smaller, but for this person, they still have the disease gene. Whereas in the siblings, there won't be a disease gene. And so this is similar to sickle cell anemia, a little bit different, and I'll get into that nuance in the next slide, but there are some diseases that do work like this. And so as a geneticist, like one of the first things we do when we're thinking about like, is a disease caused by a gene, is we'll put together large groups of families with the disease. And there are statistical methods to look at these patterns and say, are these patterns consistent with a gene that's getting passed on? And so, yeah. So as I mentioned, we look for those patterns of segregation as an indication that it could be a genetic disease. And there are two basic patterns for the way a disease like that can get expressed. The one that I showed you before is called a dominant inheritance, and that means that there is just one mutation. And if you have that one mutation, you'll get the disease. There's another type of inheritance called recessive inheritance, and this is actually how sickle cell works. And so with sickle cell, you actually need a disease mutation on both of the chromosomes that you get. And so that means that each of your parents has a mutation in the sickle cell gene, for example, but because the way that gene works, having a single mutation doesn't cause disease. So it's only when those two mutations come together and this person is getting one disease mutation from one parent and the other from the other, then they end up with disease. And so you can see some differences in the patterns here. With a dominant disease, one of the hallmarks is that you see disease in every generation. With a recessive disease, you see the disease skipping generations. And so usually the parents are unaffected and the offspring will have the disease. So we don't know. I will just cut to the chase right now, and I'll go into a little bit more detail about the genes that have been implicated in HS. And I'm sorry.
I was just going to say that actually goes into a question someone had, to be truthfully honest.
Sure.
Bianca D asked, what is the gene that is present for HS, and can it be turned on and off?
Yeah. So the short answer to that is we don't know enough about it. And so I'm going to get into a little bit more of the complexities now, why the answer is no, there's not one gene that we can turn on and off. We know a little bit about the genetics, but there's a lot more work to be done, is the short answer to this. So like I said, this is a very particular circumstance where there is one mutation causing the disease. It either needs to come together with a second mutation from a parent, or sometimes just by itself it can work. Let me just jump ahead. I mean, one of the nice things about having these patterns that are known and worked out is that when I'm doing statistical analysis to figure out if there is a disease gene and where that is, you can use like our knowledge about segregation to kind of anticipate what the genotypes are of family members. And you can use that in mathematical models to figure out where in the genome the disease is coming from. And so for instance, with a recessive disease or the sickle cell-like disease, you know that the affected offspring has to have 2 copies of the mutation. That means that each parent has to have 1 copy of the mutation, and that 1 copy was present in at least one of their parents. Whereas with a dominant disease, you know that every affected person has 1 copy of that, and that the unaffected parents likely have 0 copies of it. And you can use that for discovering genes. So here is where some of the complexity comes in. So what I just was explaining to you is how disease arises from what's called a single gene disorder, or a monogenic, meaning a single gene in a fancier word. It's also called a Mendelian disease because we have these patterns that were figured out by Gregor Mendel hundreds of years ago. This is not very common in the population. And so people estimate that maybe like 8% of the population has a monogenic disorder, has a disease caused by this. Sorry, my house is, I live in a very loud neighborhood.
Okay, I do too. Mine would be doing that right now. I'm not sure. It's a slow day for them.
Yeah, so where was I with this single gene? Oh yeah, so even in a disease like breast cancer, Alzheimer's disease, diseases like that that are considered common diseases, somewhere between 5 to 10% of people will have a single gene disorder causing that. So in a group of 100 people with HS, most of the people will not have a single gene trait. That said, there's still other ways that the genome can contribute to increase your risk for having HS, but it gets a little bit more complicated. So in this case, you don't see that very specific pattern of Mendelian segregation, but you do see that the risk within a family is greater than within a population. It's just not a clear-cut pattern. Let's see, so most of the people who have a disease will have it because of a multifactorial issue in their genome. And that also means that there are a lot of people walking around with disease variants that we find in multifactorial traits that don't have the disease. So it's not as strongly correlated as it is with that. Yeah, and so like I said, the multifactorial diseases, what we know about them is that there are hundreds of genetic variants in your genome that can work together to increase your risk of disease. And so we used to think of these as like different types of diseases, but what's become apparent with all of the sequencing that we can do now is that for every common human disease, you'll have some people who have a single gene trait and some people who don't have a single gene disorder, but they'll have risk in their genome arising from some subset of genetic variants. So now that subset of genetic variants that's contributing to it is the same subset in every person, but there'll be different contributions, I should say. Anyway, so this is just a visual image, what I've been saying before. If you take a group of people with disease, a couple of them will likely have like a single disease gene that we can say this is what's causing HS. We can predict the risk in their children. If it's dominant, it's a 50% risk. If it's recessive, it's a 25% risk. And then for most of the people, we won't be able to get this level of information. And a lot of the work that I do is figuring out where these genes are, how they're interrupting biology, and how we can use information about that to help manage disease or learn something about the biology of it. Let me see. And so there's different experimental approaches that we use for it to get into what is known about the genetics of HS. So there are 4 genes that are considered to be single gene causes of HS that have been published. And so you'll hear like geneticists talking about NCSTN is an abbreviation for a gene called nicastrin, psenin, presenilin 1, and then this PTSPIP1 gene. So these 2 genes were discovered from that first method that I told you, where some dermatologists and scientists saw that there were these segregation patterns in these very large families. So here you can see all of these people are related. The segregation pattern, so people who have HS are in the dark circle, people without it are in the light circle. And because there were so many relatives coming from the same family, they were able to use those statistical methods and find out that the portion of the genome that was traveling through the generations contained this gene in some families, and in other families contain this gene. So we do know some genetic knowledge. The problem is that it appears that most people with HS don't have mutations in these genes. Somewhere between some studies have found 0% of their patients have it. Some studies have found 5 or 6% of their patients have it. So there's some variability, but it doesn't seem to be very prevalent. The other thing that I think is important is that if you look at the way HS is presenting in these people, it's not a typical presentation, in that you're also seeing a lot of breakout folliculitis on the back and on the neck and the scalp, and that's not always typical with HS. And so my feeling is that this represents a distinct type of HS that is not very prevalent or common. And so most people with HS will not have mutations in these genes, and it's not helpful for figuring out their family risk or what's causing their disease. The other thing I have to say about these genes is that when you can link a disease gene to a segregation pattern like this, that provides really strong evidence. So I showed you how over the generations that part of the genome that has the disease mutation gets smaller and smaller and smaller. So you get really confident that this is the part of the genome with it, and this is the disease gene. There's other types of ways that people can identify these genes that aren't as robust to chance. And so the evidence just isn't as strong for these 2 genes. And so one of the things that bothers me actually is, I have dermatologists who write me every now and then saying that they have a patient who went and got their genome sequenced, and they saw they have a mutation in this gene or in this gene. And what can they do with that? They want to screen, can they do preventative measures to make sure it doesn't get passed on? The problem is that there's lots of variants in these genes that don't cause any effect. And our confidence that these are really causing HS is not so strong. And so you really have to take this information with a grain of salt and not rely on it. So basically what I would say, what we know for sure about HS is that some people can have mutations in these 2 genes, and that can cause disease. A lot of my work is focused on identifying more single gene disorders because I think that there's going to be a lot more genes, and I would personally like to get to the point where we have, so in inflammatory bowel disease, for instance, there's been a lot of genetic work on that. There are 67 genes that can cause very severe IBD that starts very early on in life. And so now when an infant or a child presents with the IBD symptoms, so severe stomach cramps and diarrhea and failure to thrive, they can go and get those genes tested to see if a mutation is causing that disease. And if they can identify that gene, it gives them information about what kind of treatments they're going to respond to. So among those 67 genes, they affect different pathways or different processes in the cells. And so you can imagine that if there are 2 different pathways that can be perturbed, you would want 2 different medications to address those 2 different problems. And so there's a lot of really interesting work done in IBD where they have all of this knowledge. And I think that we can get to that point with HS by designing these really large, well-designed genetic studies. And so that's a goal of my research program, is to try to identify more of these so that we can get to the point of really identifying people who have a monogenic etiology and learning something about what we call disease heterogeneity, or different causes of HS. I'm early on in my research program, so these are goals that I have. Jasmine, did you have a question?
Yeah, it was actually interesting because a patient, Angela Dresselhaus, was asking, because they are a identical twin. And so they said that one treatment might work for one twin and then the other might not work for the other. So, you know, despite one twin struggling with weight and the other struggling with nicotine disease severity, has been similar. Is there a genetic explanation for the different pathway, the different path to treatment for HS?
Yeah, and so I would say that that comes back to this issue, is that when you're dealing with what we call polygenic risk, or lots of these variants with individually very small effects that come together, in these cases, you have a lot of opportunity for the environment to interact with this genetic background. And so that is one way that identical twins can have different experiences with disease. If there's slight differences in their exposures, for instance, is one way, or epigenetics can also influence this. And so, for instance, in this person's genome, they have, let's say, 100 risk alleles for these polygenic risk alleles, but epigenetics comes along and shuts off a bunch of them, right? Then that would protect them from disease, or it turns on some that weren't being expressed, right? And so there's lots of opportunity in this case, which is what most of the people have in terms of genetic contributions to disease, for there to be these subtle differences.
So Shelby Demers was asking, she said, I've heard mixed things on whether more men or women are more prone to having HS. Is there any recent data speaking to that?
Yeah, and so definitely in the research that I do a lot of research in hospital records up until now, because I've just, I've been building my own cohorts, but while that takes time, and while I'm building my own studies, it's easy to access medical records to do studies in there. And definitely in medical records, you do see more women with HS than men. I think that the caveat is that women are more likely to go get treatment than men. And so it really isn't clear how much that is contributing to it and how much of it is a gender difference. I mean, that said, there is also evidence for some people, the hormones influence it. For some people with HS, there are strong correlations, right? With getting your period or being pregnant can make it get much worse or much better. And so in cases like that, you know that there's some sort of hormonal influence for some people. And if that's the case, that could explain why it's thought that more women have disease than men.
Mm-hmm.
So it does appear that, but it's not really well understood, I would say. And there's definitely opportunities for more rigorous studies about why that is.
Yeah, because I've seen also some studies that have said that it's more aggressive in men, because men have, there was a man who was studying at USC who actually was a football player, and he worked at the LA Times, and he had it under his armpits. He also had it on his head, his groin. He had it all over, and so I know that some women do experience it on their face and their heads and things of that nature, but does it present differently and more aggressively for men than it does for women?
So there is some literature suggesting that. So some people have done some studies showing that it does, HS does look different in men than women, that men are more likely to get it on their backs and buttocks, whereas women are more likely to have it in their armpits and groin. Again, the limitation is that those studies have been pretty small. And so it's really not clear exactly what's going on with that. That's one of the things that, like, as we start to discover more of what we call the genetic architecture, or all the players in the genome that could be contributing to it, we can actually address those questions more rigorously.
Mm-hmm. Is there a specific, so I know that you talked about the specific genes that they have found that could correlate to HS. Could you go over those one more time, just the names of them? Because some patients had a question.
Yeah. So the other thing that I have to say about this is that these are the two genes that there's pretty strong evidence that for some people, if you get a severe mutation in this gene or this gene, then that is likely to be a cause of HS. Most people will not have mutations in these genes. For these genes, the evidence is more tenuous. You'll find some reports of it, but people really don't understand which mutations are causing it and how those mutations are working. The other thing is, I mean, one of the main reason why I study genetics is because once you have a gene, you can start thinking about biology that's being disrupted, and you can really understand like the pathways that are causing the disease. That work hasn't been done yet in HS. So even though these genes were identified more than 10 years ago, people still don't know how it is. Like, why is it when you lose one copy of this gene, how does it get you here? People don't know. Is it because this gene is should be functioning in skin cells? Is it a dysfunction of immune cells that aren't responding correctly? They don't even know those basic questions from it yet. So there's so much that we don't know yet about HS.
I think this can lead. So Kimberly, I do want to say that unfortunately we don't know exactly why the HS would skip someone in the family. So for an example, my mom, on my mom's side, my cousin has HS. So I got HS, my brother does not have HS. So we don't understand just yet why, for an example, in my situation, my brother didn't get it, but I have it. My mom doesn't have it, but I do. Like, and nobody else on my family. So this is a distant cousin. This is like a so none of my aunts or my uncles, anything like that have it. I'm just the only one, along with my cousin. So I did want to say that before we get into the next question, which is, have any jumping genes been identified with genetic studies in patients with HS? I'm not sure what that jumping genes are. So would you be able to speak to that?
I'm not sure what that means.
Okay. Michaela, if you could clarify what jumping genes are, I would love to see if we can answer your question. So Leslie asked, you mentioned that this family, do you believe similar genes can reveal severity or intensity of HS?
You know, that's a good question. And without knowing the genes, it's hard to say. I mean, you can find samples that have been published for other diseases where there are some genes that do make disease worse for people. But we just don't know like if that will be the case with HS or not yet. We need to do more basic gene discovery before we can start answering questions like that.
Mm-hmm. And I think too, that also goes into the next question of, you know, do identical twins have HS, with HS have the same genetic mutation? Because we don't understand exactly what those genes are yet. I mean, correct me if I'm wrong, but I would have to say that we're not exactly sure if they have the same gene mutation just yet.
Exactly. Or, you know, we have to remember with a disease like HS, there are what we call environmental causes of it. So chloracne, I don't know if anybody's heard of chloracne, but it's a type of skin disease that actually comes from exposure to a poison. There is a very famous case a few years ago where the Russians poisoned somebody and he developed this chloracne, which starts out like HS. He developed boils in his groin. And and it looks very, very similar to HS, but it's because he had this poison in his body, and as his body worked out the poison and got it out of the system, it went into remission. And so that's an extreme example, right? Where a high exposure to a toxin causes a disease that looks like HS, but you could extrapolate that to imagine that some people who do have HS won't necessarily have disease mutations causing it, but for some reason are more sensitive to something in their environment. And so it's really important to remember that there are limitations to the way the genome can influence disease. My work is all focused on getting the genes that cause it, but that's not to say that that's not saying that everyone who has it is going to have a genetic cause for it, that there's other things in the environment. You can think about that like with diabetes too. So diabetes exists at higher rates in places where there's a lot of sugar in your diet, right? And so we know because the prevalence is higher when there's a lot of sugar in the community, that some of the people who have diabetes in that case have it because of their exposure to sugar and not because of a genetic mutation. So one of the challenges with my work is figuring out like how to separate out people who have a genetic cause or contribution from people who have an environmental sensitivity.
Yeah, and I would assume that economics has a huge part to play in that, because when you think of diabetes, for an example, there are a lot of communities that live in food deserts where there are a lot of processed and sugar producing foods. So that increases the rate of diabetes because of the access and not necessarily because of anything internally that's going on.
Yeah.
Absolutely. So I feel like we've answered this next question, but I'll just say it. I'll just say it. So they were asking if there's a difference between sort of like a diagnostic test for, someone who has HS in their family for someone who is the first person to present. And I'm guessing that the answer to that would be that we don't have all of that information just yet to figure that out or to identify that. But this is stuff that, people like yourself are working on.
Yeah, so I mean, that is the goal of my research, is to find those genes so that we can have diagnostic assays to help figure out if it is a genetic cause and if that genetic cause suggests a particular treatment would be beneficial. So at this point, the answer is no, there's not a diagnostic, but there are ways to participate in genetic studies to contribute to the research to try to understand what those genes are, so that hopefully in the not so distant future that we will have some diagnostic tests, like the ones that are available for IBD, for instance.
Is there, I'm sorry, go ahead.
No, it's okay.
Is there a book or some type of research that patients can dig into to understand a little bit more about genetics and how it might influence HS that you would recommend that's digestible? Because I know a lot of clinical studies have all of this medical language.
The jargon that scientists can't get rid of. That is a great question. That is a really great question, and I can't answer it, but I will definitely give it a thought. It's one of my goals at some point, once I get my research program up and off the ground, to start creating material like that, because one of the problems is that there's a huge divide created by this language difference between scientists and regular people. So I think there's definitely a need for it, but let me think about it and let me look into some different options for that. And if I find anything, I will forward it to Jasmine and she can post it on one of her things.
That sounds
but what
good.
I'm really excited, what I haven't told you guys yet is a little bit of, so what I didn't tell you is that I came to school to do this doctorate training kind of like after I already had a career. I'm a little bit late getting started in my research career, which means that I'm early in my research program. I do have some preliminary results I wanted to share with you that kind of illustrate one of the reasons why I do genetic studies. It's the type of genetic study when we're looking at the polygenic architecture. We're not going to get to a diagnostic from that, but we can learn about the biology of the disease. And so this is from the very first genetic study that I did for HS. And it was an attempt to investigate the polygenic risk alleles. And so what you're looking at now is I tested a set of 6 million genetic variants throughout the genome to see if any of them were associated with HS. Is there an allele that's more common among people with HS than people who don't have HS? Now, unfortunately, this was a relatively small study for a genetic study. Because we're doing so much testing, we have a threshold that we decide beforehand that if this threshold is not passed, then we can't say for sure that the association didn't happen by chance. And so the sad part about this is my very first study did not give us any rigorous information about genetics, but nonetheless, we did find something interesting because I figured, well, I'm waiting to build up my cohorts and do bigger studies that will have the power to get over this threshold, this red line here. But in the meantime, can we learn something about the disease by looking at the association? And so because of all the sequencing and mapping that's been done, we can go into the human genome and know exactly where in the genome these genetic variants are. And so here we're zooming into the genome. Here we're looking at across the whole genome. And here we're looking at just this little bitty tiny portion right here on chromosome 16. And when you zoom in here, so this stack of dots is the same here. They're just spread out because you're zoomed in. And you can see that there are some genes in this region, right? A bunch of genes. And so we asked the question, are any of these, the first question we asked is there a connection between this allele and these genes? And what we found was that this allele actually influences the expression levels of all of these genes, these 5 genes here. What that means is that if you have this risk allele that's associated with HS, you'll have less gene expression or lower levels of protein coming from these 5 genes, right? So we thought that was kind of interesting. And then we wanted to know, are any of these genes relevant for HS? So we went into some other data where another research group took skin biopsies from people with HS. So they took a biopsy from the HS lesion, and in that same person, they took a biopsy, a sample of their skin, just away from the lesion. So the skin looked healthy, but same person. And then they measured the levels of these genes in their skin. And what they found was that in the lesions, you see lower levels of this protein, right? And so now not only is there this genetic association where the allele is more frequent among people with HS, we linked the allele to protein levels, and we showed that these protein levels are changed in the lesion. So it's all of this evidence suggests that that there is some biology that we can learn just from studying these non-significant results. Now, the really cool thing is that it turns out that this gene has been studied in other models and systems for other reasons. But what we know about this gene is that it responds to physical force. And so in some cells, if they get squeezed for some reason, this gene will get turned on or off in response to that. Why that happens, we don't know, but I think that that's super interesting because we know that HS happens in skin that has a lot of mechanical friction. And so we're just starting to get clues now that, you know, that maybe one of the reasons why it happens in the skin fold is because there's some protein that's responsible for responding to the mechanical friction. And this could be one of those genes. So actually I'm collaborating with somebody at NYU who studies genes and how they affect model organisms. So she can actually go into a mouse and make a mutation in this gene and start characterizing to see how it affects the skin to have this gene mutated. And so in this particular example, this is not information that's going to help any one patient, but it could provide us information to understand why HS is happening for some people. What is one of the pathways or one of the mechanisms that's causing it?
Absolutely. And there are a few questions in here that I actually am going to save for Dr. Dave Louie tomorrow, or for Jillian, who is our nutritionist and dietitian. So the questions about the antibiotics in the gut, the birth control, and the PCOS, I'm going to save for the other days. So I'm writing them down. So make sure that you tune into those days as well, because I'm going to ask those questions to those panelists, just because I want to focus on genes for this specific session. So going into the next question, which I think is a really good question, Angie is asking, in your research, have you found that HS affects people differently of different races, thinking about our genes and how you mentioned the way the genes are expressed? If so, the research should, is there any research that talks about any treatments that are best, I guess, specifically for people of color? I don't know if you can speak to that.
Yeah, and so again, because we don't know that much about the genetics of it, it's really hard to say that. And I can think of one example in the literature where there is a genetic variant that is very common in people with African ancestry and very rare in people with European ancestry. And that gene causes, that particular genetic variant causes kidney disease. And so people think that that variant explains why there's differences in kidney disease between African Americans and Americans, and European Americans. And that is the only example that I can think of in all of the genetics literature that I know. I would say that what is more often the case is that even if the genetic variants are different across different ancestries, which we know they are, that they're all converging on the same biology. I don't think that there are biological differences that account for differences in disease. And so I will say, yes, there is a lot of literature saying that HS is more prevalent among African Americans than European Americans. But what we can't say, and I don't think has been addressed well with any scientific study, is how much of that disease severity is due to barriers to healthcare access or utilization. And so if you're Black in America, you're more likely to have a lower socioeconomic position. You're more likely to not have access to health insurance. You're more likely to not have access to healthcare, which means that you're living with this disease while it's flaring up and getting worse and worse. And so by the time you go seek medical care, you're in a much worse position than somebody who has the economic means and the access to go as soon as they start, developing symptoms. And so I don't think that that can explain all of the differences, but I think that it's probably contributing to some, and nobody has really addressed that, I think, with satisfaction. So the short answer is we don't know, and the longer-winded answer is we definitely need to study it more.
Absolutely. Bianca D was asking where she can find more information about the Russian poison. Would you mind spelling that for them?
I'm gonna have to Google it, which I will do really quickly to see if I can find it. And then I can post it in the chat. But that's the end of my talk. I just want to thank quickly, these genetic studies are really large endeavors. So the genetic study that I just showed you that did not have enough power had 600 HS cases in it, 600. What we really need to get to is 10,000, 20,000. So that's going to take a global effort, but it's going to take a lot of people locally also investing in these studies. So I have a lot of people to thank for the studies that I have been able to accomplish in terms of both funding sources and collaborators that are helping me to do this work.
Angela was asking about the 23andMe. They said, can I trust 23andMe raw data that says I have the NCSTN gene?
No, so everybody has an NCSTN gene. Everyone has that. Lots of people have mutations in that gene and don't have HS. There is a very specific type of mutation that has to be in that gene that is linked to the disease. So it's not enough for them to say that you have the gene or you have a mutation in that gene. Somebody, a doctor would have to look to see the type of mutation to say if it is or not likely to be.
Absolutely. And then also, I think wrapping up, have with HS does that mean? So an anonymous attendee asked, if two people with HS have a baby, does that mean the baby will get HS worse and at a younger age? We kind of talked about that a little bit earlier. And so essentially, we are not very sure exactly how it's going to present in the child that you have, or if it will. And then we also are unsure of the severity of what the child will experience if they were to get HS. So those are unknowns at the moment. And I don't know if you wanted to add anything else to that, Dr. Lynn, but that's kind of the summation of what we were talking about.
Yeah, absolutely. That we just don't know enough about the genetics now to say how it's going to affect the risk in your child. And so we can hypothesize a little bit and say, like, let's say it is a dominant gene, there's still a 50% chance that your child won't have it, right? But we don't know that it's a dominant gene. Chances are, right, only 8% of people would have a disease like caused by a single gene. So chances are that it's not. And in those cases, it's not possible to predict it at this point. And then, again, if we can say that, okay, if it is a recessive disease and both people have it, your child would only have a 1 in 4 chance of having it. So, so nothing is deterministic with genetics. And then estimating the risk of it or the likelihood of it, we can't do until we know more about the genetics.
Following up on that last question, and then I have one more question, we're going to end. So Angela was asking sort of like a follow-up to the other question about the NCSTN gene. They were also saying that there was present the PSEN1 and PSTPIP1. Would you be able to speak to that as well?
I'm sorry, what is the specific? So what I had mentioned about those the PSEN1 gene and the PSTPIP1 gene is that we have less confidence that they are a cause of HS. And so again, both of those genes have a lot of variants in them in healthy people. And so I think we need bigger studies to more definitively say whether or not they could be causing it. So there are some reports right now, but there's just not evidence showing, not to the same extent that those other two genes are.
Absolutely. Ending the summit, I want to thank you so much for this amazing in-depth explanation about genetics and HS. This was wonderful, and I know the attendees got so much information from today. I just wanted to ask one last question before we end, and I'd love to end the summit, off on a hopeful note. So concerning genetics and HS, what do patients have to look forward to in the future for genetic studies, whether that be in the next, couple of years, the next couple of months? What can they look forward to?
Yeah, so I'm actually, I received funding to launch a genetic study to start reaching out to people just through the internet to collect some data that's needed for my analyses about their experience with the disease, and then to send out saliva collection kits. So similar to what 23andMe does, where you would you'd fill out the survey, spin the tube, and send it back to me. And then I'll include that in our genetic studies. So, I would say, I think that there is a lot that we can accomplish in a very short amount of time, just because of all the people who came before us and all the work that's been done for these other diseases. It gives us a good roadmap for what we need to do to really get some answers that will be helpful. So I'm hoping within the next couple of years, we'll have more information about which genes that will be able to actually do some clinical sequencing like they can do for breast cancer right now, or IBD, to get to that point. But so I would just say to keep your eye out for ways to help out with these genetic studies. And, in my research program, I mentioned that it is early, but as I said, it's super important to me that the research gets back to the people that it affects. And so I'm hoping to have a website of my own set up at some point for that kind of communication with people. so that, you know, for me to get my research published in a scientific journal, I have to use a lot of jargon or it won't get there. But that does a disservice to all the people who really need that information, right? And so I think it's something that's very important to me, is finding ways to like establish communication that's understandable.
Absolutely. Thank you so much. And thank you to everyone who tuned in for the first day of the HS Awareness Virtual Summit. We have so much more planned for this week. Tomorrow specifically, we have Understanding HS, which is the basics of HS. So a lot of the questions that you all had, maybe regarding to gut health, nutrition, and things of that nature, we can get some of that tomorrow. And then some of the more heavy hitting questions about diet and supplements, we're going to get into the next day with Jillian. So make sure you tune in tomorrow, because we have Dr. Stephen Dave Louie, who is a dermatologist at Wayne State University in Detroit, Michigan, who will be speaking to the basics of HS. So use the same link that you used today to access the session tomorrow, and we will see you right here at 7 p.m. Eastern Standard Time, 4 p.m. Pacific Standard Time.